Good evening, everyone, and welcome to this year's Croonian Lecture. The Croonian Lecture is one of the oldest annual lectures of the Royal Society, and it's our most prestigious lecture in the life sciences. It was conceived by William Croon, who was one of the original fellows of the Royal Society, and among the papers left in his death in 1684 were plans to endow two lectureships, one at the Royal Society and the other at the Royal College of Physicians. His widow later bequeathed the means to carry out the scheme and indicated that the bequest was for the support of a lecture and illustrative experiment, which sort of shows the times, for the advancement of natural knowledge on local motion, or, conditionally, of such subjects as is, in the opinion of the President for the time being, should be most useful in promoting the objects for which the Royal Society was instituted. So that has been taken to mean significant work in the life sciences. And the first Croonian lecture was delivered in 1738, and the lectureship today is accompanied by a medal and a gift of £10,000, which I'm told has been wired directly, so the check is in the mail. So this year's, we're really very fortunate to have in this year's Croonian lecturer, Professor Jennifer Doudna, who is also a foreign member of the Royal Society, for her outstanding structural and functional studies of RNA and ribonucleoproteins, and for elucidating the molecular mechanism of the CRISPR-Cas9 system and developing it for genetic engineering. So to tell you a little bit about Jennifer, Jennifer Doudna graduated from Pomona College in California after having grown up in Hawaii. And she then went to work with Jack Szostak for her PhD at Harvard, where she became interested in RNA catalysis. And continuing that interest, she went on to do a postdoc with Tom Cech at the University of Colorado in Boulder. And that's where she began her studies on the group 1 intron ribozyme. And to understand it, people had been, it was clear that you would need a structure to really understand details of the catalytic mechanism. But nothing bigger than tRNA had been crystallized in the previous 20 years. And this, even the construct that, well, I don't know what that text says, but anyway, you better just listen to me. Maybe it's my accent. Anyway, even in order to identify a construct, she had to do a considerable amount of biochemistry to identify a construct that was both interesting and compact enough to crystallize. And she got her initial crystals and then moved on to a faculty position at Yale University, where she solved the structure of the P4-P6 domain of the group 1 intron. And this was a big milestone for several reasons. It was several times bigger than tRNA, which was 20 years old at that time. And so it completely opened up new understanding of RNA folding and tertiary interaction motifs. It also shed light on how group 1 intron could take part in catalysis, although that work came later. Another important advance that this work made was phasing of large RNA structures. And this is something many people don't realize, but the compound that Jennifer and her team used, osmium hexamine, was actually then the basis for phasing much larger RNAs like the ribosome. And so really it paved the way for a breakthrough in the crystallography of the ribosome as well. Now, at Yale and then at Berkeley, she worked on many other important ribozymes, on the mechanism of silencing, on translation and its control, And these were all areas at the forefront of RNA biology. At some point, she became interested in the CRISPR Cas9 system, and I'm not going to tell you anything about it because it's the topic of her lecture. But I will say it is a remarkable example of how interest in what might seem an esoteric topic, bacterial immunity to phage, is revolutionizing not only biological research, but potentially human health. And the work has now revolutionized the field of gene and genome editing. And as a result of her very distinguished career, she's received many honors and awards, including membership in the National Academy of Sciences and as I mentioned, foreign member of the Royal Society. She's received, among others, the Gardner, the Japan, and the Breakthrough Prizes. She was named one of the most 100 influential people by Time magazine. And as if all this weren't enough, she's also founded several companies to use this technology to improve human health. She's been active in the public sphere, especially in discussion of ethics around genome editing and the sort of social and ethical implications of it. And finally, I should say she's the author of a very highly readable book called A Crack in Creation, which really lays out her journey into this field. So without further ado, I welcome Jennifer to give her lecture.
Well, good evening, everyone. It's a great honor for me to be here, and I'm really delighted to see many friends and colleagues, and of course to have that very generous introduction by Venki Ramakrishnan, one of the people who has inspired my career in structural biology and thinking about the function of RNA. As you heard in the introduction, I've had a long-standing fascination with RNA molecules and what they do in biology, but the story I'm going to tell you tonight really has two different major players. One of them is DNA, sort of thinking about the structure of the double helix and how this molecule encodes the information necessary for life. It's also, honestly, the way that I got excited about doing biochemistry back when I was a young student growing up on a little island in the Pacific, Hawaii, where I learned about the discovery of the double helical structure. And that book that described that discovery by Jim Watson was the book that inspired me to start thinking about a career in a field that would allow me to discover molecular structures. I was fascinated by the idea that somebody could do that kind of work and understand the structure of molecules. And the other major player in the story tonight is bacteriophage that infect bacteria. So this is a cartoon showing a phage landing on the surface of a bacterial cell. And like all viral infections, this involves injection of genetic material, in this case DNA encoding the information to make a virus, into the cell. And it was through the study of a process that cells use, an RNA-guided process to protect cells from this kind of infection, that it was possible to understand the molecules involved and recognize their potential to be useful for a very different purpose. And that's really what I want to talk about tonight, is really the work done by our lab in collaboration with the lab of Emmanuel Charpentier to figure out how bacteria fight off viral infections and in the process use enzymes that can be programmed for a very different purpose, namely for genome editing. And so what I'm going to do tonight is I want to start out by telling you a little bit about CRISPR biology and how we got interested in this, as you heard, esoteric area of biology, and then how our work to understand the molecular process involved led to harnessing of this technology as a genome engineering tool. And then I hope you'll indulge me a little bit. I want to tell you a bit about research that we've been doing in my lab over the past 6 years to understand the molecular basis for this process. And then I'm going to, towards the end of the talk, I want to turn to the topic of applications of genome editing and the exciting opportunities, as well as I think the really fundamental challenges that we face in harnessing this tool going forward. But let me start by describing CRISPR Cast as a bacterial adaptive immune system. So the existence of these systems was unknown to science, really probably until the late 1980s. So all of the beautiful research done on bacterial genetics in the 50s, 60s, and 70s hadn't uncovered the existence of a way that bacteria fight viral infections that allows acquiring resistance to viruses. And that was because these are largely not operable in the kinds of bacteria that scientists have studied in the lab. They occur mostly in bacteria that are growing out in nature somewhere. And so it was really through that kind of discovery by a few laboratories around the world that it became clear that bacteria can acquire DNA from viruses and use it to protect themselves from future infection. And here's how it works. And this is summarizing research from several different labs. But what happens is that here's the membrane of a bacterial cell, and here's the inside of the cell. And if this bacteria is infected by a virus, which injects its genetic material, if the bacterium has a CRISPR system, it has a place in the genome where it can store small pieces of viral DNA. And those pieces are stored in a very particular way. This is called a CRISPR locus, where there are a series of DNA repeat elements shown by the black diamonds that flank these unique bits of viral DNA shown by the colored boxes. So it's a very distinctive pattern that was first recognized by computational biologists who were looking at bacterial DNA sequences and noticed these things. They didn't know what they were doing at the time. And these reside next door to a series of genes that encode CRISPR-related proteins, or Cas for CRISPR-associated. So these are Cas genes that encode proteins that turn out to interact with these RNA transcripts that are generated from the CRISPR locus, shown here initially as a precursor molecule that gets processed into small pieces of RNA that each include one of the virally derived sequences. And when those combine with the Cas proteins, they form surveillance complexes that can search through the cell looking for pieces of DNA that match the letters in this 20 nucleotide stretch of the CRISPR RNA molecule. So it's a really beautiful system, and you might notice that by these colors that each of these proteins is programmed with a different RNA sequence that can recognize a different type of virus, a different viral DNA. And then once that recognition happens, the proteins are able to use base pairing between the RNA and the DNA to capture that piece of nucleic acid and allow it to be cut by the Cas protein. And this leads ultimately to destruction of the viral nucleic acid and protection of the cell. So it's a very interesting pathway, and we were fascinated. I was initially fascinated by this because of the role of these RNA-guided proteins and trying to understand what that might tell us about the evolution of these sorts of processes more broadly, not just in bacteria, but also in other kinds of cells, potentially. And so we started working on this, and one of the things that's very interesting about these CRISPR-Cas systems is that they're very diverse in nature. And this is a cartoon that shows an attempt by the lab of Eugene Koonin and his colleagues to categorize the CRISPR-Cas systems according to the types of proteins that are found encoded next to the CRISPR arrays. And so each of these little boxes is a different Cas gene, and they're color-coded by what they do in the system. And quite broadly, one can separate these into two classes called Class 1 and Class 2 that are distinguished by the number and types of Caste proteins that contribute to the pathway. So the class 1 systems have multiple Caste proteins that are involved in protecting cells using an RNA-guided mechanism. But down here, the class 2 systems each involve just a single large protein that has the ability to protect the cell in an RNA-guided fashion. And so when I met Emmanuelle Charpentier at a conference in 2011, she told me about research that her lab was doing on a bacterium that had this type of CRISPR system here with a single protein called Cas9 that was implicated as an RNA protecting enzyme. And at the time, nobody knew the function of this protein, and we thought it would be fascinating to figure out how it works. And so that was really the question that we set out to address together was what is the function of this fascinating enzyme, Cas9? And so in research that was conducted in our 2 labs across oceans and different time zones by 2 very talented people, Martin Jinek, a postdoc in my lab at the time, and Chris Chylinski, a graduate student with Emmanuelle, these guys figured out that Cas9 is an amazing enzyme, shown here in this cartoon as the blue blobby structure, that has the ability to hold on to double-stranded DNA at a place in the sequence matching the 20 letters in the CRISPR RNA molecule, this molecule right here. And when that interaction occurs, the protein makes a cut in the DNA, and it uses 2 separate active sites to generate a blunt double-stranded break. So in bacteria, that's a terrific way to cut the viral DNA and initiate the process of degradation. Now, Martin and Chris Chylinski figured out 2 other important things about this enzyme. One is that in nature, it requires a second piece of RNA called tracer, shown here in red, that interacts with the end of the CRISPR molecule to provide a structural handle for Cas9 binding. So it's really a dual RNA-guided protein. And they also figured out that this targeting mechanism requires not only a match to the CRISPR RNA sequence, but it also requires that this site be next door to a small motif in the DNA called the PAM motif, which for this enzyme that we started working on is a GG dinucleotide, so two Gs in the DNA. And that turns out to be important, as I'll explain shortly, for the mechanism of DNA recognition. Now, Martin Jinek, being a wonderful biochemist, and he actually had originally done research in Kiyoshi Nagai's lab here in the UK before coming to my lab to do his postdoctoral work, he figured out that these 2 RNAs could be linked together to make a single guide form of the sequence that would have the RNA targeting portion on one end and the handle for Cas9 binding on the other. And the really exciting thing about this development was that we realized when Martin did experiments with this single guide RNA that it made the system trivial for programming Cas9 either in vitro, so biochemically, or in cells. And the reason is that now this is a 2-component system with a single protein, Cas9, and a single piece of RNA that could be programmed by changing this sequence in the RNA to cleave any desired DNA sequence. And by using it in that way, it was possible to introduce double-stranded breaks into the DNA of eukaryotic cells, plant and animal cells, that would trigger DNA repair. And this sort of recognition, was one of those sort of really precious aha moments that we all live for in science, where you're doing experiments in the lab and you don't quite know where it's going. You're trying to answer a particular question. And then when you see the data, you realize that the data tell you something unexpected, that or explain something about biology that you couldn't have imagined. And for us, that was the case here, because what had been going on in other fields sort of for the last couple of decades before we did this research was a lot of work on understanding how DNA is repaired in eukaryotic cells, in plants and animals, where double-stranded breaks, rather than leading to quick degradation of the DNA and cellular death, typically are detected in the cell and repaired by pathways that include non-homologous end joining, where the ends of the DNA are ligated back together, sometimes with a very small change in the DNA sequence, or by a process that involves homologous recombination, where a piece of DNA that has a sequence matching the sequence of DNA that was cut can be integrated into the DNA during the repair process. And many scientists appreciating this, this process recognize that you could actually use this to trigger changes to the genomes of cells by introducing a double-stranded break at a desired place in the genome. And the big challenge was, how do you do that? Not so easy. And so there were a number of technologies that had been developed involving primarily programmable proteins that could be engineered to bind to particular DNA sequences. And these could work very robustly for gene editing, but they turned out to be difficult enough to deploy as a technology that it had not been widely adopted. And the thing that's so exciting about CRISPR-Cas9 is that this system is essentially very trivial to utilize by people that have molecular biology training. And so it became a tool that was almost immediately adopted very widely for applications in all sorts of different kinds of cells. I'm going to show you one quick little video here. This was a movie made by Janet Owasa to illustrate how we imagine this CRISPR-Cas9 enzyme functioning in a genome editing context. So here we are zooming into a eukaryotic cell. So the DNA is in the nucleus, and it's highly compacted into chromatin, so wrapped around these histone proteins. And this programmed enzyme, so the Cas9 protein with its guide RNA, will search through the DNA to find a sequence that matches the sequence of the guide RNA. When that match occurs, this protein has the ability to unwind the DNA helix, cut the two strands of the DNA, and then hand those ends off to repair enzymes that will fix the DNA, here with quite a bit of artistic license, integrating a new piece of DNA sequence. And amazingly, this enzyme that evolved in bacteria, and to our knowledge does not exist in eukaryotic cells, nonetheless works very well when it's engineered to go into the nucleus like this and make double-stranded DNA breaks. So I'm going to come to some of the applications of this towards the end of the talk. What I want to turn to now is to tell you a bit about the research that my own lab has been doing to understand how this actually works. We've been sort of fascinated by this question of DNA recognition, all the way from, how does this enzyme find a DNA sequence and allow an RNA strand to interfere with the double helix by opening up the DNA to form an RNA-DNA hybrid, all the way to how does this actually work in the context of a living cell. And I'll show you just tonight some of the experiments that we've done and data that we have that address these sorts of questions. And I wanted to start by just pointing out that one of the really interesting things about Cas9, this enzyme that emerged early on, is that this is a protein that it's effectively a helicase. It has the ability to melt apart the strands of DNA at a position matching the sequence of the guide RNA. And how does that work? We've been trying to figure this out. So this is a 3D printed model of a Cas9 guide RNA complex bound to a double-stranded DNA substrate. It's a model that's actually based on an actual 3D crystallographic structure that was solved in Martin Ginek's lab at the University of Zurich. And what I'm showing you here is the protein with one of its cleaving domains taken away. So you can actually see the inside of the protein. And you can see that when it finds a matching sequence in DNA, it forms an RNA-DNA hybrid inside the enzyme that displaces the second strand of DNA, and in a process that we're still trying to figure out, allows this enzyme to access a functional conformation that leads to DNA cutting. And amazingly, this is an enzyme that does this without any external energy source. So it's able to somehow coax apart the DNA double helix, even in the context of a whole genome that's packaged into chromatin, by melting apart the two strands, but it doesn't use any external energy source to do that. So it has no hydrolysis of ATP or GTP that drives this process. Somehow it happens independent of that. And one of the clues to how this melting of DNA works has come from studies of crystal structures and looking at how the protein structure changes as it binds to guide RNA and then to DNA. I'm going to show you one little movie here that just morphs between different crystallographic states of Cas9 to illustrate this process of structural change. So this little movie starts with the structure of Cas9 alone, and as it morphs to the structure bound to the guide RNA here in orange, there was you might have noticed a very large rotation of this part of the enzyme that actually creates a central channel in the protein where the RNA guide is located. And then when this enzyme binds to DNA, there's an additional change in the structure that accommodates that RNA-DNA hybrid. And then finally, there's actually a sensor in this enzyme that detects the fact that it's bound to a complementary piece of DNA, and that triggers a final rotation of this active site called HNH that swings it into position to cut the DNA. So this part of the enzyme contains the cleaving residues that actually generate one of the cuts in the target strand of the DNA. Now, that final rotation that I just showed you in this video was not observed in the original crystallographic structures of Cas9 because it turns out that it only happens when this protein is engaged with a bona fide double-stranded DNA substrate. And that was possible to trap and solve structures of by two people in the lab, Fugo Zhang and David Taylor, in collaboration with the lab of Eva Nogales. And the structure I'll show you here was obtained using a combination of crystallography and electron microscopy. And I just want to show you what this looks like. So here we are zooming in to the interior of the enzyme. You can see the orange RNA with the DNA hybridized to it. And here's the other strand of DNA in magenta, sort of opened up inside the enzyme. And that structure actually positions the enzyme to put its active sites in place to actually cut DNA. And you'll see coming into place here the HNH domain, this time in green, so you can see it rotating into position. So it now it's actually placed properly to generate a break in this strand of the DNA. And importantly, this requires recognition of this PAM motif that's sitting right here next to the targeted sequence, which for this enzyme is a GG dinucleotide. And we understand now from crystal structures and other kinds of experiments that there's a binding site in the enzyme that interacts with those Gs in the DNA and somehow triggers melting of those first few base pairs that allows the start of DNA hybridization and eventually allows this entire strand to interact with the DNA. So it's really an interesting enzyme that has the ability to be programmed to interact precisely with DNA and also to sense whether it's on a bona fide target or whether it's on a not quite perfect matched DNA, which leads to binding but no cutting of the DNA. So I wanted to share with you a few experiments that we've been working on over the years to sort of address this question, which is how does Cas9 find target sites within the vast excess of non-target DNA? And the first set of experiments that we did to address this was a terrific collaboration with the lab of Eric Green at Columbia. So Eric had come to Berkeley and talked about work he was doing using DNA curtains, which are single molecules of DNA that can be sort of attached to a slide, and then one can monitor binding and binding behavior of proteins that interact with the DNA by putting a fluorescent label on the DNA and using turf microscopy. And this is what these experiments look like. So each of these strings, sort of green strands that you see here, is a phage lambda DNA molecule. So it's a 48 kilobase pair piece of DNA These are each attached on one end to a slide, and then they're extended when there's a buffer flowing across the slide from top to bottom. And then in this experiment, what we're doing is labeling Cas9 protein with a fluorophore, and then we can program that enzyme with a particular piece of guide RNA that will direct it to a site within the phage DNA. And what you can see here is that these enzymes are all lining up at roughly the place where we expect them to, according to the position of the guide RNA binding. And so it was by doing these sorts of experiments, and these were conducted by Sam Sternberg in my lab and Cy Redding, a student in Eric Green's lab, that it was possible to do experiments that established, first of all, that you could get accurate targeting in this sort of an assay. And you could see very nicely the way that these proteins would line up on the DNA according to the position of their guide RNA. And it was also possible to do things like measure the binding kinetics and how long it took for an enzyme to find its target site in the context of this 48 kb piece of DNA. And to summarize what we learned in those experiments, we figured out that, first of all, there's very high affinity product binding by the enzyme. That means that this is a protein that binds to DNA and cuts it, and then, at least in these sorts of experiments, never lets go, at least in the context of the time course of these types of assays. So there was no substrate turnover. For each enzyme that we added to these reactions, we would get one molecule of DNA cleaved. So that argues that in cells, there's probably a lot of other enzymes or proteins that have to interact with DNA to displace Cas9. Secondly, we found out that binding occurs first at these PAM motifs. So at least one of the ways that this enzyme operates is by searching for these GG dinucleotides in a sequence of DNA and slowing down only upon encountering those. And of course, they would be quite abundant in a typical genome. But nonetheless, this provides probably the first constraint for this enzyme to localize to places where there could be an actual target sequence for DNA recognition. And then finally, we figured out that target binding somehow triggers Cas9 catalytic activity. So this is an enzyme that is poised to bind to DNA, but only once it's engaged with an actually a piece of DNA that has a match to the RNA guide is it triggered to activate and cut the DNA. And that's probably important for survival of bacteria. They wouldn't want to have an enzyme let loose in the genome that's making inaccurate breaks and potentially leading to cellular death. So with those sort of observations in hand, we set about doing a series of experiments that involved using dye molecules on the surface of the Cas9 protein to monitor conformational changes. We were really intrigued by the possibility from these comparisons of crystal structures that this enzyme might undergo a big structural rearrangement upon DNA binding. But until you actually detect that in solution, we want to be a little bit skeptical of, is that really how it's working? And so to test that, this was originally the work of Sam Sternberg in the lab. So what Sam did was to look at the various crystal structures of this enzyme and engineer into the protein sequence residues that could be modified chemically with pairs of dye molecules in such a way that we could detect fluorescence resonance energy transfer, or FRET, between the dyes. And so what I'm showing you here is an example where we have a Cas9 protein. You can see the difference in the inactive versus the active states of this enzyme. And when we have dye molecules positioned here and here, they're too far apart to give a FRET signal when the protein is in the off state, so not actively engaged with DNA. But once this active site swings into position on a piece of DNA, then they become very closely positioned. And so Sam Sternberg originally did these experiments in bulk solution and got some very nice data that were consistent with the conformational changes detected by comparing crystal structures. But we wanted to do experiments that would give us even more insight into the behavior of individual molecules. And this led to a great collaboration with the biophysicist Ahmet Yildiz at Berkeley and his student Yavuz and my student Janice. So these two students teamed up to tether Cas9 molecules to slide surfaces that would, in a way that would allow them to do single molecule fret measurements. And in this type of experiment, what was done was to use a piece of DNA that would base pair with the end of the guide RNA molecule in Cas9. And this protein, of course, has the dye molecules on the surface, so we can monitor fret changes. And then we flew into the system different pieces of DNA that have the PAM site as well as a target sequence. And the target can either be a perfect match to the guide RNA or contain different numbers of mismatches. And then we can monitor the fret changes that occur in Cas9. And I just want to show you a couple of the types of data that we got that started to really explain, I think, a lot about how this enzyme functions. So in one of their experiments, Yavuz and Janice were able to compare the fret signals that we observed for individual molecules as a function of the type of DNA that was added to the system. And so what you're seeing here are, and again, these are single molecule fret experiments, so we're binning the fret signal that we get from individual molecules of Cas9 according to the type of DNA that was added to the system. And so when we have a perfectly matched piece of DNA that matches the CRISPR RNA, these molecules show a very high fret signal. And again, that's consistent with these dye molecules being very close together. So we're getting a very high signal in that assay compared to what we see for RNA only here. This is where we have no DNA added to the system, and the molecules are all in a very low fret state. So they're all in the off position. And then what's interesting is what happens in between. So when we have DNA molecules that have even a single base pair, we start to see population of this intermediate state monitored by fret. And you see that the larger number of mismatches in the DNA, the more we see population of that intermediate. And with this and some additional experiments, we were able to conclude that this represents a conformational checkpoint of Cas9. It's really a state that the enzyme has to pass through to become fully activated. And it's really influenced by the degree of base pairing with the target DNA molecule. Furthermore, we could do experiments of this nature where instead of tethering the Cas9 protein, one could actually just tether the substrate DNA molecule to the slide, flow in the protein with its fluorescent labels, and monitor the rate of change of the protein structure as it interacts with DNA. And what you can see here at the bottom is that when we use an on-target piece of DNA that has a perfect match to the guide RNA, there's very rapid occupancy of that high fret state. So very quickly, within a few seconds, this enzyme grabs onto the DNA and snaps into position to cut it. Whereas if we use a piece of DNA that has mismatches to the guide RNA, we see that it takes a lot longer. So these are 10 to 20 seconds here to even start to occupy that activated state. And for the most part, most of the molecules remain over here in this low fret inactive state. Again, pointing out that this is an enzyme that has a sensor that is detecting the degree of base pairing matches between the RNA and the DNA. And then finally, I wanted to show you that this activation is dependent on divalent metal ions. So this is doing these same types of fret experiments where we have different types of divalent ions included in the reaction. If we use no divalent ions and we add a chelator to grab all of those ions and make them unavailable to the enzyme, you might see here that all of these enzymes are locked in this conformational intermediate state. But as soon as we add just a pinch of magnesium, 10 micromolar in this experiment, we start to see population of that activated state, and that grows in as we add different types of divalent ions. But interestingly, that's independent of the enzyme being able to actually cut DNA, because if we use a version of Cas9 that has a mutation in the active site, it nonetheless can occupy that activated state. So it really suggests that binding and this conformational change are separate from actual DNA cutting, which has been actually important for using this as a tool for DNA recognition in different types of genome manipulation experiments. So just to summarize what I just told you, this is an enzyme that goes through this conformational checkpoint that allows detection of base pairing between the DNA and the RNA. And in data that I won't show you tonight, we were able to use this understanding of the mechanism to make mutations in and figure out where this DNA sensor is in Cas9. and mutate it so that it makes the enzyme more accurate, even more sensitive to mismatches in DNA, which is proving to be an important aspect of genome editing, where we want to ensure that edits are made only at the desired position in a genome and not anywhere else. So one other thing I wanted to share with you about the Cas9 mechanism is thinking about how we take this biochemical understanding and apply it to thinking about the way this enzyme operates in the context of a living cell. And so from the work that we did with Eric Green's lab using those DNA curtains, we figured out that our data were most consistent with a model where this enzyme, rather than sliding along DNA to find a binding site, is instead binding and releasing DNA very quickly and slowing down only at PAM sequences to allow initial base pairing between the adjacent sequence of DNA and this guide RNA. And if there is complementarity there, then this starts to form a structure that eventually looks like this if there's full complementarity and leads to DNA cutting. But how does this work in the context of a genome? And so I had a wonderful student, Spencer Knight, who came to the lab a few years ago, and he wanted to try to answer this question by thinking about how Cas9 would be able to interact with structures like this, namely nucleosomes that are the components of chromatin in eukaryotic cells. And so the questions were, how does Cas9 deal with chromatin structure? How does it handle big genomes? And how does it deal with nuclear organization, sort of the higher order architecture of genomes? And we certainly don't have answers to all of those questions yet, but got some important initial information by conducting experiments in which we included a halotag on the end of Cas9. And so this is actually doing experiments with the catalytically inactive form of the enzyme with a halotag on the end. So this allows coupling of a very small fluorescent dye to this protein. And importantly, one does this in the context of a living cell. So by expressing this fusion protein in a living cell and then adding this dye molecule that will go into the cells and react with this halo domain over here, we get a fluorescently labeled Cas9. And I'll just show you a couple of results using this in super resolution microscopy experiments to visualize the behavior of Cas9. And these are in living mouse cells. And so one thing that we noticed right away was that when we compared the kinetic behavior of Cas9 proteins that were programmed with a nonsense guide, so they don't recognize a sequence in the genome, compared to what we see for Cas9s programmed with a guide RNA that recognizes an abundant sequence in the genome, a repetitive element called a sign, we found that, and you can probably just see it visually here, that these individual proteins moved a lot slower in the cell when they were programmed with a sign recognizing CRISPR RNA. And you can plot this over here. This is just the log of the diffusion coefficient plotted against the frequency, number of particles with that behavior. And so you can see that we get very rapid diffusion when there's no DNA recognition going on, but there's a lot of these particles have much slower kinetic behavior when they start binding to the DNA. And so by doing these sorts of measurements, it was possible to calculate, at least roughly, how long it takes for Cas9 to find target sequences in the cell, and also to do experiments like this, where we could actually visualize the behavior of these labeled Cas9 proteins in the context of cells that had an additional label in a protein that binds to heterochromatin. These are parts of the genome that are really, really compact. And one question that we had was, would this protein with its rapid kinetic behavior be able to get into these highly compacted parts of the genome? And you don't see much when I show it to you like this, but when we compile all of these images, what you can notice is that these bright regions are the heterochromatic parts of the genome, and you can see that these colored paths show the kinetic behavior of individual Cas9 proteins. Much of the time, the protein is outside of heterochromatic regions, but we see forays into these structures as well. And that's consistent with the observation that we can get gene editing in these heterochromatic parts of the DNA. It just takes longer than it does in the rest of the genome. So it's really a remarkable observation that this enzyme is able to somehow insinuate itself into even these highly compacted portions of the genome, which perhaps speaks to the dynamic nature of even these very compacted parts of a typical eukaryotic genome. So in the last few minutes, I just want to turn to the challenges that are ahead for taking this system and sort of the understanding that we and others are developing about how the function of this protein allows it to be a programmable enzyme that initiates genome editing, and thinking about how we're going to take it from where this technology stands today to solving real problems in clinical medicine, in agriculture, and in other areas of biology, including synthetic biology. And I wanted to point out a couple of things. So I think, first of all, that when we think about challenges, especially for therapeutic applications of genome editing, I really see three major ones. One is delivery, so how we can introduce these genome editing molecules into cells and tissues in patients, how we control the way that DNA repair happens after the DNA is cut, and finally, how we deal with the ethical challenges that come up when we think about certain types of applications of genome editing, especially in humans. So one of the things that's happened over the last 6 years is just the remarkable acceleration of research in the biological sciences, enabled by a number of technologies, but including Cas9. And I just wanted to show you this plot. So this is I recently was writing a review for a journal on CRISPR Cast, and I sat down on a Sunday morning to kind of go through PubMed, and I told my husband, oh, this will probably take me a few hours to do this. And several hours later, I was only gotten through 2 months of the first part of this year. There were just so many publications. And so right now, this is probably now even out of date, but about 8,400 entries. And this is just to give you a sense of what's happened since, sort of really over the last 6 years, where labs worldwide have started to employ gene editing to do all sorts of experiments. And we now have sort of a library of all of the different types of cells and organisms that have been edited using CRISPR-Cas9. This is actually just a partial illustrated map here of these different types of organisms, but it's really been remarkable to see the kinds of interesting biology that's been enabled by having a tool that allows easy manipulation of genomes. And I just wanted to tell you about 5 areas where I think there's some really exciting applications that are happening, and just give you examples of some of the things that I think are most interesting in these different areas. And there's lots of things I could have picked, but these are some of the things that I've been thinking about recently. And I want to mention research, sort of fundamental research, healthcare, therapeutics, agriculture, and diagnostics. So in research, I'm just going to give you two examples that illustrate the way that having a genome editor at your fingertips as a scientist has really opened the door to doing new biology. So this was a picture that I got from Claude Desplan at New York University and his students who have been using CRISPR-Cas9 to make genetic changes in butterflies that allow them to study in genetic detail the wing patterns of these butterflies. And this was something that until there was an easy way to edit genomes was possible to do only at the level of observation and just looking in nature at what kinds of aberrant types of wing patterns would occur. But now they have a tool that allows them to manipulate those genes exactly and ask questions that couldn't have been addressed in the past. There was also a recent article, this is actually from The Guardian, talking about the work that Svante Pääbo and his team are doing to understand the evolution of the brains of Homo sapiens. And so they've been looking at the genetics of Neanderthals, as some of you may know, and experiments they're doing currently involve growing small organoids of neuronal cells in a dish where they can actually introduce into a human-derived organoid a segment of DNA that's taken from the Neanderthal genome. And they're focusing on 3 different genes that they think are important, perhaps, for the development of certain types of neural functions in Homo sapiens, to ask whether they can recreate that in these settings in the laboratory. Again, an experiment that would have been impossible to do without a tool that allows site-directed gene editing. In terms of healthcare, so there's lots of focus on biomedicine, but one of the areas that I think is actually also very interesting is thinking about ways that we might be able to have an impact on healthcare in the future that don't involve doing any gene editing on humans. And that is, this is an example here where George Church and others are interested in asking whether they can develop pigs that have been genome edited in different ways to remove endogenous viruses and to make their organs more human-like that would allow these animals to be great organ donors for human patients. In our own lab, we've been thinking about how we might be able to employ the CRISPR-Cas9 enzyme to target disease genes. And we've actually been working on a gene for Huntington's disease. This is a well-known gene that involves a mutation that was mapped actually back in the 1980s. So we've known about this mutation, but it's been impossible up until now to offer these patients anything really other than palliative care. And so this is showing an experiment done by a postdoc in my lab, Brett Stahl, who was able to modify the Cas9 protein with its guide RNA so that this protein has cell-penetrating properties. And by injecting this into the brains of mice that are engineered genetically so that we can observe the editing reaction by simply watching the cells turn red when they get edited, we can actually observe that we get really significant volumes of brain tissue that gets edited when we do these kinds of injections. And we're actually working now towards developing this as a tool that will work in a larger animal model and looking for even better ways to get penetration of these proteins so that we can edit enough cells that we would have a therapeutic benefit in principle in patients. And I want to point out that in agriculture, there are just so many opportunities right now with gene editing. I think people are very excited about what's going to be possible in the future. This is a great example from the lab of Zach Lippman at Cold Spring Harbor Laboratories, who was able to use CRISPR-Cas9 to generate strains of tomatoes that carry much heavier fruit yields than the unedited plants, something that he told me would have been either difficult or impossible to do previously, and certainly now can be done within a matter of weeks by engineering just the gene that is necessary to change for this very productive type of tomato plant to be produced. And finally, I just want to mention that in recent work by our lab and several others, it's been possible to harness these CRISPR enzymes for detection of nucleic acid sequences. And this is taking advantage of the properties of some Cas proteins, not Cas9, but other sort of related RNA-guided enzymes, to recognize DNA or RNA molecules and then trigger cutting of a fluorescently labeled piece of DNA or RNA. And by doing this type of reaction, it's possible to detect the presence of viral DNA, even potentially DNA associated with cancer cells, tumors, in very small volumes and without using any kind of fancy equipment like a PCR machine. So there's a lot of excitement right now about the potential to be able to detect DNA and RNA sequences using this simple kind of technology that might be applicable in a point-of-care setting. And I just want to close by circling back to sort of some of the broader ethical issues. And for me, I think it was really back in 2014, sort of my evolution, my own thinking in this whole field started with, being incredibly excited about the potential of the technology and all the things that it was doing and all the papers that started getting published and people harnessing it for different applications, to realizing quite early on that this technology was going to be, could be used for different kinds of editing experiments. And I wanted to just point out that fundamentally, we can divide cell editing into somatic cell applications, meaning non-heritable changes that are made to DNA. They're not passed on to future generations, versus what happens when we conduct germline genome editing, where changes become heritable. So they affect the entire organism and its future progeny. And so this was being done from very early on in various kinds of animals and plants, but it became clear quite quickly that this would work also in primates. And people started to ask the question, what about human germline editing? Would this be something that people should do, either for research purposes or for clinical applications? And of course, that quickly led to people imagining CRISPR babies and all the sorts of things that one might like to engineer into our kids. But I think there is a very real possibility that in the future, certainly not today, but at some point in the future, that this will become a reality. It will become possible to introduce changes in a targeted way into human germ cells, whether it's eggs or sperm or embryos, that become part of the person and become part of their future lineage. And it's a really profound thought, if you think about it, because it really means that we now have a tool that allows us to control our own evolution, if we want to. And the question is, what do we do with a technology like this? How do we manage it? How do we handle it? I don't know the answer to that question, but it did seem very clear to me that it needs to be publicly discussed. And so together with a number of colleagues, many of whom are in this room, we got together and started talking about this. And there was an international summit held on this topic back in 2015, and another one to be held this year in November in Hong Kong. And the first meeting actually led to publication of this report on human genome editing that discusses the science and the opportunities as well as the challenges of this technology, especially in the context of human germline editing. I think this is a great start, but as we all are aware, this field continues to move very, very quickly, new technologies and sort of improvements to the technology happening all the time. And so there continues to be a need to discuss this and encourage more public contemplation of how we take appropriate stewardship of this technology going forward. And I just want to end by pointing out that, as I said, the field continues to move very quickly, and one of the things that's happening is that, as I mentioned at the beginning of the talk, these CRISPR systems are very diverse in nature. So it's possible to find new ones, and this is taken from a publication of ours together with Jill Banfield from last year, where we found several new examples of Caste proteins that are RNA-guided enzymes that have no similarity to Cas9, but they function in a similar fashion. So we know that there will be opportunities to find these types of enzymes, and they may have new functions that allow new kinds of applications. So it continues to be a really exciting field in terms of fundamental discovery, as well as thinking about applications and how to make sure we have responsible use of these technologies. So I'll just conclude by pointing out that RNA-guided gene regulation occurs in a variety of biological contexts. So this is kind of the CRISPR Cast system is kind of the latest example of RNA-guided control of genomes. You may be familiar with RNA interference in eukaryotic cells. So this is clearly a very important way that cells can recognize nucleic acids and manipulate them in different ways. We know that applications of genome editing will depend on both delivery and control. And when I say control, I mean both chemical and societal. And then finally, that fundamental research will continue to explore new CRISPR systems and what comes after CRISPR. And so we just, I think we're at a really exciting moment in biology when this technology, coming together with others, are putting tools in the hands of scientists that we've never had before and that really open the door to doing all kinds of research and engineering and applications that would have been almost unthinkable just a few years back. So with that, I'd like to conclude by acknowledging a terrific group of people in my laboratory. So I mentioned many of these students along the way, and I also want to point out that we've had terrific collaborators as well, beginning with Emmanuel, but also a number of people at Berkeley, and I didn't mention Joel Palevsky, but he's been working with us to develop Caste proteins for human papillomavirus detection, something that we're hoping to be able to do eventually in point-of-care clinics. Finally, we couldn't do anything without our funding agencies. And this is where I really like to point out that I think the importance of supporting fundamental curiosity-driven science really can't be overemphasized. It's so critical that we have opportunities to work on problems that we think are interesting because, as you can see in this story, there's a certain serendipity to scientific discovery that really can't be predicted. And with that, I'll close, and I'd be happy to take questions if you have them. Thank you.
Well, thank you very much, Jennifer, for that real tour de force that covered everything from fundamental sort of bacterial phage infection and going all the way to structure and function to a wide range of applications and ethical issues. So it really shows a tremendous breadth. And you talked about serendipity, but it does remind me of Louis Pasteur saying that chance favors the prepared mind. And it was that ability to sort of see all those connections that's really very remarkable. So it's now my, before we go to questions, I think I will do this first because I might forget later. And so it's my pleasure to give you a scroll and a medal that goes with the lecture. So thank you very much. So the floor is now open for questions. So if people have questions, could you?
So, you said you don't have the answer to the correct ethical way to use this very powerful technology, but perhaps you have an opinion. What is it?
Where do I start? I think it's, first of all, critical that we work to develop a consensus among scientists about how to use this. I think that many of us, and certainly many people before me, have concluded that it's very hard to imagine putting in place regulations that would be adopted globally and respected globally necessarily, and how would you enforce such a thing? So I think next best to that is encouraging a scientific consensus about use of technology that provides a framework for how we deploy it going forward. So that's really the goal, I think, of these kinds of meetings and reports right now, is to encourage that. And then I think the second piece is really to have an open discussion so that people who are not scientists are not blindsided by what happens when technologies start to be applied in different ways. So I think those are two important pieces to how to proceed.
So I had a question. You know, from your FRET studies and other biochemical data you showed, it looked like even when there were mismatches, there was some significant signal. But the actual off-target sort of, you know, effect is reportedly much smaller than I would have thought from that signal. Is that because the sort of homologous recombination is pretty inefficient, and so it sort of amplifies the real site compared to the, I don't know, what is the
I think it's actually, what I think it's due to is, I think it's a function of using a minimal amount of Cas9 in cells so that you get, you really minimize the number of off-target cleavage events that occur. And that's really been underscored, I would say, by a lot of the studies that have been done with enzymes that are modified in different ways to try to make them more accurate. And what we often find when we test these biochemically is that you just lower the overall level of activity. So I think that you can achieve that in a cellular setting by just reducing the concentration of Cas9 in the cells. And so I think as people have gotten more sophisticated about how to deploy this in those sorts of genome editing applications, we're now working typically with limiting amounts of Cas9, so you really minimize off-target events for that reason. Yeah.
So is the technology getting to the point where it's safe from an off-target point of view, or how far away are we?
Yeah, I think the answer is yes, in the sense that what I've seen for data coming from primary cells, so not cancer cells cultured in the lab, but cells that are from primary tissues, plant systems, and also human cells, is that when you do deep sequencing looking for off targets using from cells edited using a minimal amount of Cas9, it's actually remarkably accurate. So it's not that easy to find off targets. And the other thing that's done now that wasn't appreciated early on is that you can design guide RNAs to largely avoid most of the close matches in the genome and avoid off-targeting for that reason. So I think it's getting to a point where you can actually envision clinical uses. It's not to say that we don't need to continue development, but I think it's good enough now that people are really contemplating how you might employ it in a clinical setting.
Hi, Jennifer. I wanted to ask you, what's your opinion on ribonuclease targeting chimeras? Do you think that they offer a quicker route to therapeutics than the CRISPR technologies?
Are you talking about RNA targeting versions of the Caste proteins, or?
No, ribonuclease targeting chimeras.
Ribonuclease, sorry, I'm still
targeting
missing.
chimeras.
Ribonuclease chimeras?
Ribonuclease targeting chimeras.
Ribonuclease targeting chimeras.
Yeah, so we've had quite, so I work for a company called Frontline Genomics, and we've had some interesting articles about this new technology. Maybe we could talk about it afterwards.
Maybe we'll talk about it afterwards, yeah.
No problem.
Hello. Could you tell me what's your opinion about using CRISPR to modify people for fun? Like,
I don't recommend it.
I was wondering with the talk you did there, but the template part of RNA is 20 bases, and with the starting motif, I take it that it's always 20 bases. Is that enough to actually accurately edit the human genome, i.e. 3 billion base pairs? You've already said that you can get off target because of the not matching completely, but in terms of the template, I take it is always fixed at 20 bases, or can you change that for accuracy? Can you do anything with that?
Right, so it turns out 20 bases is just about right to get recognition of a single sequence in the human genome. Although again, as I mentioned, you can adjust the sequence of the guide RNA to ensure that you avoid targeting, closely matched sequences in a genome like that. But people are also fooling around with making longer guide RNAs. And so far with Cas9, that hasn't worked. And the reason is that the protein protects 20 nucleotides of guide RNA. And when we make extensions on the ends of the RNA, they get nibbled off in the cell, probably by nucleases. So it's sort of a architectural explanation for that. But there may be natural systems that use longer guide RNAs. We've been working with one recently in the lab that's using a 23 base pair guide. So there may be natural systems that use longer RNAs, and there may be applications where that's advantageous.
So is there any research to try and work out whether that's advantageous, or people are just finding different, you said there's different versions to Cas9, different molecules. Are you looking at the accuracy of, because obviously if you want to do editing, you want it to be as accurate as possible.
Of course. Yeah, I think that's certainly a topic for future investigation. Currently, what I can tell you is that it's one of these crazy serendipitous things, but this enzyme that Emmanuel Charpentier and I started working on turns out to so far be still largely the best at this kind of gene editing. And we think we have a biochemical explanation for that in work that I didn't talk to you about tonight, but we think that this enzyme is a remarkably good helicase. It's very good at melting DNA compared to a lot of the other natural variants of Cas9, for example, found in bacteria. And so it's not as trivial as one might think to just take new RNA-guided enzymes and apply them for gene editing. They don't always work right away, and I think that has to do with, at least in part, with the biochemical behavior of these proteins.
Thank you.
Hi, so I'm from the Science Policy Research Unit at the University of Sussex, and I'm particularly interested in the biosecurity aspect of CRISPR and how it relates to dual-use research of concern. Do you think that there's a hype surrounding CRISPR being used as a biological weapon and how it can be used by non-acting state, non-actor, non-state actors. Yeah.
Do I think there's hype? Yes, I do, because I think no offense to any media that are here, reporters, but I think that those are the kinds of articles that get attention. But I also think that it's an important topic and something important to think about, because there, of course, is potential for this technology to be used inappropriately and unethically. So this is one of the goals, I think, of the meetings that are ongoing, is to discuss what those are and try to mitigate against it. And again, it's a real challenge, because it's, as you might have appreciated from this talk, this is a technology that's widely available. It's easy to get a hold of. There are now kits sold on the internet for high school kids if they want to, make green fungi and things like that using CRISPR-Cas9. So it's, it's just, I think it's a topic that is going to continue to require careful attention. And the good news is there are lots of folks like you and your colleagues that are thinking about this and discussing it and trying to figure out appropriate ways to control it.
Hello.
Hi.
There's been a lot of debate, especially in the EU, are these products GMO, not GMO, et cetera? How should they be regulated? What do you think is an appropriate framework and way to think about this technology and the products of this technology and how they should be addressed?
I was fascinated to learn when I got into this that the way GMO is defined is different in different countries. So in the US, GMO is defined according to the actual product that results from the manipulation, whereas in Europe, the GMO is defined according to technology that's used to create something. So the same product that in the United States would be considered non-GMO because edits were made to the genome that didn't involve introduction of any foreign DNA, for example, would in European countries be considered GMO. So it's, it's one of these kind of crazy things. So I think that it really brings home a couple of things. One is thinking about how we actually define something like that, and it's a bit of a technical thing. It's hard to explain that to people that don't have a technical background, what the difference is. But what I like to point out as well is that, especially when we're thinking about GMO plants, effectively everything we consume is GMO because it's been bred typically to have certain properties that we find desirable. And plant breeders traditionally have used methods to introduce changes to DNA that involve either radiation or chemical mutagens that introduce all sorts of random changes to DNA. They select plants that have desired properties, but they don't control for what other changes in the DNA are coming along for the ride. And so I think the really powerful thing about CRISPR-Cas9 is that this is now a technology that gives plant breeders a tool for making a precise single change to a genome, or multiple changes but in desired places that don't bring along any other mutations in principle. So I think this is really what people need to appreciate, and this is what is, I think, under active discussion, is how we understand that technology, explain it, and then regulate it appropriately given the way it works.
There were a couple of questions over here.
Hi. You touched on the other nucleases. Given that the vast amount of research goes into the standard Cas9, do you ever think from a commercial point of view that we'll see the other nucleases enter the clinic?
Absolutely. I do, yeah.
One over by the end there.
Thank you for a wonderful lecture. It's fascinating, but I'm particularly interested when you said none of this requires energy. Am I right? That you actually said all these reactions don't depend on...
External. Doesn't require an external energy source.
External.
Yeah.
And can you explain that a bit more?
Yeah. So a lot of enzymes that manipulate DNA, and especially those that melt it apart, use an external energy source. So that means they use molecules of ATP or GTP to drive that kind of DNA unwinding. And what's amazing about this protein is it doesn't do that. So it has no need to hydrolyze ATP to drive this melting reaction. It does it somehow through a series of conformational changes and probably taking advantage of the thermodynamics of RNA binding to DNA.
That's fascinating. Can I just go on there? And what implication would that have for the evolution of prokaryotic and eukaryotic cells if this doesn't require an external source?
I'd be hard-pressed to sort of fit that into the sort of overall evolutionary argument for these organisms. I think that it's possible that, you know, in bacteria that, you know, these enzymes have to work fast. They have to be very, very fast. And they have to be very efficient at DNA recognition. And that perhaps precludes having to also bind to a second molecule of ATP to hydrolyze. But I'm speculating.
Thank you very much.
Well, we have time for just 2 more questions. So, one here. Up.
Thank you so much. That was a really interesting talk. I was hoping to live tweet it, but I was completely indulged in the experience. My question actually follows on from my colleague from the Science Policy Research Unit. So you discussed that there's been a meeting in which scientists and other people have talked about sort of the ethics surrounding the usage. And you also touched on the fact that perhaps in the future we could design our babies or whatever. So my question is, in future discussions that you have surrounding perhaps governance or non-governance, what sort of ethical codes or codes, so what level do you talk about in terms of discussing the ethics of usage? Because you mentioned that it would be less about enforcing governance or regulation and more about an open discussion within scientists. But do you think that needs to extend to perhaps state members who could benefit or use the technology for their own agenda? So what level of discussion do you think is necessary, from your perspective, in order to ensure that, you know, this technology isn't used for bad things, basically?
Well, I guess I think about the example of in vitro fertilization. So I'm old enough to remember before there was in vitro fertilization and then after, right? And so in my house, you know, growing up, I remember my parents debating when the first, you know, test tube babies were being born, and my parents, you know, debating this, and is this right, and should people do this? And, you know, and I think that was common in a lot of, you know, a lot of families and groups. And then what happened was that, you know, Louise Brown grew up and she was normal, and other babies were born and they appeared to be normal, and this gave an opportunity for couples that were infertile to have kids. And there was such an incredible desire to do that industry really developed rather organically. And I suspect that what we'll see with gene editing, you know, could take a similar path in the sense that, you know, there's already in vitro fertilization going on, and people do pre-implantation genetic diagnosis to figure out if embryos have particular genetic traits. And so we could imagine that going a step further, where at some point it becomes offered to parents in those sorts of clinics this opportunity to do gene editing. And, you know, and so the question becomes, you know, how should we try to control that process or let it happen organically? Should we try to engage governments in this discussion? In my experience, I don't know what it's like here in the UK, but in my experience in the US, I've been to Congress maybe 3 times, and we only have one. Let's just start by saying we have only one PhD scientist in our entire Congress. Shocking. But you know, in my experience, so the congressional members that I've met with are typically not sophisticated scientifically, but they also are very, very interested in this technology. So they really want to understand it. So I am sensing you know, sort of a desire on the part of our government officials, at least in the US, to figure out what's going on. Whether that will mean they actually put in place responsible legislation, I wouldn't want to gamble on that one. So I think, you know, again, I keep coming back to the fact that I think scientists really need to be deeply engaged in this. We need to be discussing it publicly. We need to be sharing our information. And we all just need to, you know, try to work together to figure out the best path forward it's a complicated topic, as you know, and it's a really important one, and it's one that many people are very interested in, which is a good thing.
Okay, so I realize this is an endlessly fascinating topic, but I'm afraid we have to bring the proceedings to a close. And I want to finally conclude by thanking Jennifer Doudna again for really a splendid talk covering a huge range of